Hey everybody, hope you're having a happy Friday. I want to wish everyone a happy weekend. I'm going to do a little bit of training here after last week on efficacy of our multivariate coherence approach. I want to dive a little bit into an intro on aspects of connectivity because that's very important in understanding the work that we do. So let's talk about that a little bit today. So there are three basic types of connectivity as outlined in Sporn's Networks of the Brain book and by others. It includes what we call structural connectivity, which is defined as a set of physical and anatomical connections in the brain. This is the hardwiring of the brain. These are the highways, okay? The only way we have of seeing this information is either at autopsy, and that's the way we've done it for centuries until something called diffusion tensor imaging was created, where you can actually image those networks by studying the transfer of water molecules in an MRI machine, and you can actually see the pathways. This is the structure of the pathways, not the functioning of the pathways. The next thing is what's called functional connectivity, and these are patterns of deviation from statistical independence of units that are remote from each other but are related to each other. The basis of this is typically a time series where their relation is taken as neuronal coupling, and by relation, usually we're talking about a correlation. It's taken as coupling, and it's taken to imply that there is some quote unquote connectivity across those regions, but there's no causal relationship, effect, or interaction. It's just a correlation, what they're doing similarly at two different locations. And when we talk about this type of functional connectivity, we can measure that with any type of neuroimaging technique like functional MRI, where you can see activity in remote locations, such as the inferior parietal sulcus and the posterior cingulate cortex. And as they covary on this graph, when they're correlated, when they move in similar ways, then they're shown to be correlated. And when they move in opposite ways, they're said to be not. Okay. That by itself doesn't necessarily mean that one is causing the other. It just means that they're doing it at the same time. This can also happen in PET scan correlations. And of course, our understanding mainly comes from EEG correlations. So you can have this with any number of EEG recordings, but these lines down here will typically represent a value of coherence between two locations. And that's what we refer to as functional connectivity. Again, there's no implication that one is causing the other, just that they're correlated. Then we move into what's called effective connectivity. Effective connectivity means that there are network and causal effects between neural elements. This is really what we're looking for. We want to know what causes what and how we can influence that. This is inferred through statistical techniques such as time series analyses and statistical modeling that assesses causality and interaction. This requires complex data processing and modeling techniques such as independent components analysis, partial directed coherence, and Granger causality. These are all taken as multivariate autoregressive models. And in the simplest way of looking at it, it's like a multiple regression technique over multiple locations. So now it's not just pairs that we're looking at. We can look at multiple points and even source-directed points simultaneously. So here's an example of what that looks like in real life, where in these images, you not only see the locations, but you see arrows connecting them directly. There will often be a value represented, the degree of connectivity or degree of cause. For example, explaining 50% of the variance in a statistic, and you'll often see an arrow pointing in a certain direction. And so we can measure the influence or cause in one direction, and we can measure it in the direction going back. So you get reciprocal causes. And as we get into the software that we use, I'll explain more about that and how that works. But that's what can be measured in these effective connectivity models, and it's very important. That gives us the real information we want to work with. Well, many people that work in EEG and neurofeedback don't have access to this type of connectivity, and they use what's called coherence. Again, coherence is functional connectivity. The Granger causality model that we use is the effective connectivity. Okay, for a long time, the standard method in establishing relations between signals has been what we call cross correlation or coherence. Again, directionality cannot be ascertained. There is no reciprocal causality or any causality at all. And you can have other factors that are influencing the correlation, which may have nothing to do with one area impacting another. And in fact, some of the error rates of paired coherences go as high as close to 50%. So let's compare these approaches, effective and functional connectivity. This is an image of a diffusion tensor imaging from an fMRI study of a young woman on the left-hand side with autism and mirror neuron dysfunction, where it shows very clear dysfunction and connectivity problems going up to the prefrontal cortex compared to this image of a normal, and down into the temporal and limbic system, which would help to regulate that activity. You see a clear separation of this connectivity pattern in this clinical individual. If we look at the EEG coherence pattern of the same person, we would expect to see frontal to temporal dysfunction, and we don't see any of that, sadly, in any frequency. And if we look at a multivariate coherence image, we can see here there is a looseness or hypoconnectivity from fronto to temporal networks in multiple frequencies. So when we incorporate multivariate approaches or approaches that use multiple channels simultaneously, we get more valid and accurate information that would reflect structural or effective connectivity. But when we only use pairs, as in functional connectivity, a lot of times our estimates are very far off from reality. In this study that was done way back in 2004, they looked at a contrived experimental model where they knew the relationship between 5 different electrode points. It was a simulated model, so they already knew what they were. And basically, it was that variable number 1 would cause interaction to 2, 3, 4, and 5. And those are the only directions of the influence. So in the bivariate model that was produced, looking at correlations on this side, you can see there's a correlation here of activity between the 5 that we see, but many others as well. So you can see the actual ones by these black lines, and the others that were found to have a relationship in the red lines. But that relationship is not real. That relationship only occurs because of the correlations across multiple sites. So that is what we call spurious findings. That is, it's implying there is a relationship of connection between these areas indicated by these red arrows when that relationship actually does not exist, which is why we view coherence as such an inaccurate estimation of connectivity. But when you use a multivariate autoregressive technique, you can see we get the actual values and causes and connections that exist in this model. It's much more accurate. Another example of this was another simulated model where we go from 1 to 2 to 3 to 4 to 5, and then from 5 back to 4. So there's a reciprocal relationship between 4 and 5. And we can look at different types of metrics. So for example, typically in what we call coherence, which is here, we see that there's a relationship there's some relationship between basically every location and every possible combination. Well, we know that's not the reality. That's not what this model shows us. Otherwise, there'd be arrows between every possible connection. Okay? We can look at other metrics of coherence, what we call imaginary coherence, phase differences. And it's not until we get to this model of what's called partial directed coherence that we actually get the relationships that exist in the model as precisely as possible. Well, what is that? Partial directed coherence is the amount of directed coherence explained by the causal model. So again, it's like doing a multiple regression procedure and saying, what amount of the variance is explained by this portion of the model? So then we can target and we can determine which regions of the brain are causing activity in which other regions and have confidence that they are accurate and precise. And here, when we go from, I think it was, if we go back, we go from 4 to 5, which is this one, and 5 to 4 in the other direction, we can see there's a reciprocal cause there because they both have a relationship. And we could measure that and measure the balance of that as well, which we do in our software. Let me just show you a clinical example. So again, this is an example of an individual with major depression. We saw an example of autism with a mu rhythm problem. Now I'm going to talk about major depression. So this is what we know about research in major depression. PET scan findings show diminished activity over left frontal temporal limbic regions of the brain. fMRI data show anomalies in the left frontal, temporal, and limbic regions. And fMRI connectivity show these magenta lines across similar brain regions, maybe even going a little further back in the brain, including other areas. But there's a consensus across these strategies and techniques of what we understand to happen in major depression that's existed beyond a number of months. So what happens when we look at this with our connectivity metrics? We're expecting to see something in the left frontotemporal regions. Well, our coherence, our two pairwise coherence metrics don't show us that. And in fact, if we were to do some neurofeedback training based on this, we wouldn't train over those areas. When we look at our multivariate coherence model here, we can see a large deviation over left frontotemporal regions in alpha. And when we look at our even more precise model with ICA dipole localization and Granger causality, we're looking for the dark blue areas right over the left frontotemporal limbic regions. So that identifies for us very easily a region we would want to train in the brain with multivariate coherence neurofeedback, leading to greater increases in efficacy. And that's basically how our model works. So again, I would encourage all of you to ask questions about any of this on our sites, Facebook or LinkedIn. Start dialogues, have discussions, ask questions about any topics, recommend teachings. Again, I'm happy to go back and teach more about our efficacy studies with different populations or address any specific questions that you may have. So please feel free to interact and engage, and I'll certainly respond and do the same. Feel free to invite your colleagues, other members you think would enjoy this information, and hope to talk to you all soon. Have a great weekend. Bye-bye.